Hereby I open this academic ceremony at Maastricht University. My name is Ton de Goeij. I am the pro-rector of the ceremony and chair of the degree committee. First of all, I welcome Mr. Michael Brunner. He will defend in public his academic thesis, Development and Usefulness of Transgenic Rabbit Models of Inborn Arrhythmogenic Diseases. Welcome all members of the degree committee, in particular the two supervisors, Professor Axel Zurhausen, he is Professor of Pathology at Maastricht University, and Professor Judith Sluimer, she is Professor of Cardiovascular Pathophysiology, also at Maastricht University. I will introduce the 5 opponents later, because if I mention all those names now, you will certainly forget. Dus I do that later. Welcome you here in the aula, and I would like to welcome also all the followers of the live stream. Mr. Brunner, may I invite you to present a summary of your thesis, and I wish you success in the coming hour. The floor is yours.
Dear Pro-Rector, the members of the assessment committee, in the next few minutes I will present to you my thesis, Development and Usefulness of Transgenic Rabbit Models for Inborn Cardiac Arrhythmogenic Diseases. This is a human heart, and the heart consists of billions of cells called cardiomyocytes. And each of these cardiomyocytes creates an electrical signal. The electrical signal is called an action potential, and the action potential of all these cells together can be seen on the surface as an ECG, as we all know from a doctor's office. Changes in the action potential and in the action potential duration can be seen as a change in the QT interval on the ECG. And we know for many years that a prolongation of the QT interval, which represents a prolongation of the action potential duration, can cause arrhythmias. This is a patient I've seen 2 years ago in my emergency department. And he came in, he's 40 years old, and he had a long QT syndrome, and he came in with repetitive fainting and loss of consciousness. And we hooked him up to the monitoring, and we put an ECG on him, that's the green line, and a pulse oximetry, that's the blue line, basically a representation of the blood flow in his fingers. And while on monitoring, he developed extrasystoles, the broad complexes, and more extrasystoles, and this turns into a fast ventricular tachycardia. Tachycardia that is so fast that the heart actually stops beating, and because it has no time to perform an output or to fill itself. And you can see on the bottom line, that the blue line, that there's actually no pulse occurring anymore. And this patient fainted, and later on this arrhythmia ended spontaneously, and the patient regained consciousness. This is a classical form of a long QT syndrome. Many different proteins in the cell are responsible to create an action potential. And there are actually many more than those that you see on this slide. On this slide, it's only those that can actually prolong or shorten the action potential. Most of these proteins and channels are actually very rarely affected. It's only these 2 channels that I show you here that create 2 ion currents, IKR and IKS. These are potassium currents floating out of the cells and responsible for the repolarization of the cells. And these 2 currents are the more commonly affected in this relatively rare disease. And if you look at an action potential at the same time, again, you see in blue IKS and in yellow IKR. These are responsible for the shape of the action potential, for it actually to come down at the end of the action potential and to return to the baseline. And if one of these channels is impaired and is not working properly, then the action potential becomes longer. And you can see that both on the single cell level as well on the ECG below, you see a prolongation of the QT interval. There are rare mutations, however, that increase the current through these ion pores, and it actually shortens the action potential, and that's called a short QT syndrome. For over 70 years now, we've known the long QT syndrome, and we've known many instances of patients with long QT syndrome and arrhythmias. This is a patient from 1972, a young boy who has alternating T waves. If you look at the T waves, it's sometimes positive, sometimes negative. It's alternating, and it turns into a fast ventricular tachycardia, which ends up spontaneously. That's the classical long QT form, and we call it long QT once the QT interval is beyond 480 milliseconds. On the other hand, in the last 20 years, we have some approximately 200 to 300 patients described that have a short QT interval, and actually a short QT interval leads to the very same arrhythmias as the long QT syndrome. And this is a relatively new entity, and we define short QT syndrome as being shorter than 370 milliseconds. Having worked with mice on long QT for quite a long time, we had the hypothesis that a rabbit might be a much better model for the human disease phenotype, and we wanted to create a rabbit model that actually emulates the human phenotype. And we also wanted to analyze the mechanism of the arrhythmias, why arrhythmias occur, why they are sustained in these rabbits. And we wanted, of course, to evaluate new therapeutic approaches, because there's not a lot of therapeutic approaches right now. For this, we started with a long QT syndrome, and we got 2 mutations from 2 affected patients that we put into a construct to create long QT1 and long QT2 lines. These mutations were in the pore region of the channel, basically there where the potassium flows through. And we created these long QT rabbits, and from these long QT rabbits we got cardiac myocytes, and back again at the cellular level, we see the action potential of a wild type. That's a healthy, normal rabbit. And you see the prolonged action potential of a long QT1 rabbit and of a long QT2 rabbit below. But these rabbits did not only have prolonged action potentials, they also had a prolonged QT interval. On the right side, you see a picture of an ECG of a healthy rabbit and a prolonged QT interval of the long QT1 and a prolonged QT interval of the long QT2 rabbits. That far, we only have a long QT, but also these rabbits showed spontaneous ventricular arrhythmias, very, very similar to what I've shown you in the second slide. And it occurred that these rabbits also had polymorphic ventricular tachycardia, this fast-changing ventricular arrhythmia that actually leads to sudden cardiac death. And these rabbits had a decreased survival. In the long QT2 line, we saw that the rabbits started to die around day 200, which is the occurrence of puberty in these rabbits. And at about 1 year, about half of the long QT2 rabbits have died. On the other hand, wild-type rabbits rarely die, and long QT1 rabbits also showed no spontaneous fatal arrhythmias. So despite having a similar length of prolongation of the QT interval, affecting different currents or different channels, results in a different phenotype in the rabbit. Of course, we're also interested in the short QT syndrome, and we used a similar approach to create a transgenic short QT rabbit. We actually tried 2 different lines. One line did not work, and we did not proceed with that one. But we got short QT1 rabbits with a very similar approach, again a construct taken from a human affected with this disease. We created the short QT rabbits, and on the top you see a wild-type action potential, a normal, healthy action potential of the cardiac myocytes of these rabbits. And below you see the shortened QT interval in these rabbits. And if you look at the surface ECG of these rabbits, again you see a shortening of the QT interval. It was also that we saw in these rabbits was spontaneous ventricular arrhythmias. These are the white complexes in the top graph, which are ventricular extrasystoles. And they also had inducible and spontaneous fast polymorphic ventricular tachycardias. I'm sorry. And we saw a decreased survival in these rabbits, and this time it was not depending on puberty, but it started already in the infancy of the rabbits. It started around day 40 that these rabbits started to die. And what we also did see is we saw that action potentials in these rabbits You have a black action potential, which is a wild-type rabbit, and a red action potential, which is a short QT rabbit. They had spontaneous early after depolarization, meaning that the action potential fired again and caused these ventricular arrhythmias. So this was one of the causes for ventricular arrhythmias in these rabbits. Now that we had these models, we wanted to test a few other things, and I'm going to show you a little tiny fraction of what we actually did, because we did a lot of studies on different mechanisms and different drugs that we treated. We wanted to check whether hormones would influence arrhythmias, given the striking influence on puberty, and what we know in humans is that women with long QT2 syndrome, during pregnancy, they are protected from arrhythmias, and after giving birth to the child, the ventricular arrhythmias reoccur. They're actually very, very endangered, especially in the first weeks after giving birth. So we took female long QT rabbits, overectomized them to avoid the influence of the naturally occurring hormones, and exposed them to hormones, either as a control group to no hormones or to estrogen, testosterone, and progesterone. And if you look at the QT intervals compared to control, it was only estrogen that significantly increased the QT interval, whereas testosterone did not change a lot, and progesterone only moderately increased QT interval. We monitored these rabbits using a telemetry device that recorded the ECG, and it was only in the estrogen-treated female rabbits that we could see spontaneous arrhythmias, and we had an increase in sudden cardiac death, where actually more than 50% died during the observation period. So we concluded that estrogen prolongs the QT and induces arrhythmias in female long QT2 rabbits, and progesterone and testosterone do not induce arrhythmias, do not prolong the QT interval. Another study that we did is we hypothesized that in rabbits who are lacking one of the two repolarizing currents, an increase in the remaining repolarizing current would actually decrease the QT interval and make the phenotype more or less normal. NS1643 is one of the substances that can actually increase IKR, and we tested this in vivo and in vitro. And if you look at this graph, you see on the top line, you see the QT interval of sham-treated hearts, and on the bottom downward goes the line with the QT interval of the rabbits treated with NS1643. So it could actually decrease the QT interval significantly within 30 minutes. However, at the same time, the contractility of the heart also became depressed, and the hearts were basically not beating at all at the end of the experiment. So we made a nice ECG, but the hearts were not beating, so this is not a feasible approach, at least with this substance. Finally, and this has only been published a few weeks ago, we used Michael Ackermann's construct to perform a gene therapy approach. This construct contains a suppressor of the endogenous gene and a replacement of a functional gene. And we use this experiment first in isolated cells. And if you look at the dark blue line, you can see this is a normal long QT1 rabbit. And by exposing it to the construct in the plasmid, we could also reduce the action potential duration to the red line, meaning it's more or less the same as the black line, which is a wild-type rabbit. And we did this also in vivo in these rabbits. And in the top line, you see an untreated normal long QT2 rabbit. In the middle, you see the shortening caused by the construct after 3 weeks, shortening of the QT interval, and that's actually as short as a wild-type rabbit. So here, in a proof of principle study, we could actually show that we have a gene therapy approach for this disease. Let me conclude. The transgenic rabbits we created are a valuable tool for QT syndromes. They provide insights into the mechanism of arrhythmias, and they enable us to evaluate novel therapies on a medium-sized animal level. And I wish to thank my supervisors and my collaborators for their support with these projects. None of this would have been possible without them. Thank you very much.
Thank you for your clear summary of your thesis. The opposition will be opened by the chair of the thesis assessment committee, Professor Erik Bisse. He is Professor of Experimental Vascular Pathology at Maastricht University. Professor Bisse.
Thank you. First of all, dear candidate, my congratulations on your thesis. It's not my core expertise, but I read it with great pleasure, and it's not only very appealing from the outside, but also the content is highly impressive. So I also like to extend my congratulations to the promotion team. So I'm not only here to sort of congratulate you, but also to interrogate you. And already at page 3, I was intrigued by your leitmotif, maybe of your study, but also maybe in personal life. The quote of Guy de Maupassant stating that das Herz hat viele Geheimnisse, in die der Verstand nicht eindringt. Knowing what you know, based on your work, what do you consider the greatest challenge left after all what you have shown now?
Highly esteemed opponent, there's a lot of challenges left actually. We only started to understand. We started out to get a model that was sometimes more practical than a mouse. A mouse is not suited as a model at all for long QT, I have to admit, even though I've worked long time on it.
And based on anatomic constraints, or is that?
The mouse is basically easier to manipulate genetically, yeah. And it's very difficult to create transgenic rabbits. When we started out, there was only one publication out there which had a success rate of 1 in 1,000 oocytes that were implanted into a rabbit. And we were lucky to find somebody who brought this down to 1 in 100 implantations of oocytes. But that was a mention. And the mouse is much better explored, but its ion channels are completely different from the human. And this is opposed to the rabbit, and this is why I emphasized that we used mutations taken from humans. The rabbit's repolarizing currents are very, very, very similar to the human. And this is one of the reasons is, of course, the heart rate. A mouse has a heart rate which is 500, 600 beats per minute, and the rabbit is around 200, and the 200 is pretty close to a neonate. So the tools and building blocks are very much the same. However, we still do not understand a lot of things, how these arrhythmias are perpetuated, and how they generate. There's still a lot of work to do.
Okay, thanks. Now, you come to mention the rabbit, and you're advocating the rabbit as the ideal model for or at least one of the potentially more viable models for studying arrhythmia. So you made use of a transgenic mice, and I think the setback of a transgenic might be that you have an uncontrolled or non-physiological expression level.
Yes.
So how that work in your case for this long QT?
Well, the approach we used was to use a dominant negative approach. So we had actually an additional gene with a defect in the rabbit myocyte. And we checked that in the beginning, and we had 1 to 2 copies per cell of that. So we did not have a lot of more copies than that.
Don't titer off, yeah.
But of course, it's not the physiologic way that this disease occurs, but it was the closest we could get back then, because that was way before CRISPR-Cas that we started.
Yeah. And also the clinical features look pretty comparable to that in humans. So that's definitely a very strong case for your model. So then one of the studies I was intrigued in is described in chapter 4.1 and 4.2, where you look into the effects of sex hormones. And that's, of course, a plausible topic, considering that it's a post-puberty clinical manifestation of the model. So you look into both estrogens, progesterone, estrogen being sort of detrimental, progesterone at best maybe slightly protective even, but also at the lactation hormones like oxytocin and prolactin. So if you put that all together and look into the clinical time paths of the manifestation generally of these arrhythmia in subjects, what would you consider to be the most plausible cause? Or is there any additive or synergistic effect between these sex hormones?
Yeah, there is actually, once a woman gives birth, progesterone drops dramatically. And the balance shifts from progesterone to estrogen again.
But this, this happens quite fast.
This happens quite fast, yeah. This happens quite fast. Yeah. Plus, you have the effect of oxytocin and prolactin. And we did not replicate sort of the whole setup around a birth of a child, because that was way too complicated. We were happy to get that somewhat simulated, because estrogen and progesterone are sort of baseline hormones. They're stable, but prolactin and oxytocin are pulsatile. And to get a pulsatile hormonal exposure in a rabbit is very difficult. But what we could see is not only that estrogen is detrimental and progesterone is sort of protective, we also did see that oxytocin and prolactin both have an effect on ion currents directly, and both have an effect that actually go into the direction of being pro-arrhythmic. And this fits well with the observation in women who give birth, that they are more prone to arrhythmias in the first 6 months after giving birth. I mean, there's a lot of other influences coming in as well. There is sleep deprivation, there's arousal, which is or waking up, which is a very strong trigger for long QT syndrome. Yeah?
Well, it's pretty complex.
And stress. So all this comes together, and actually this is probably the reason why women have more sudden cardiac death after giving birth. Which actually leads me back to the first thing, is that we say it is very, very important that what we can do as a therapy, which is beta blockers right now, that women start taking up beta blockers as soon as possible after giving birth.
Okay, thank you. I give the word back. I'm fully contented with the answers. I give the word back. Still, I have time for another question. I will be happy to raise one. So one of the things I was, based on what you just told, is there any data around on postpartum effects, manifestation of long QT in women that don't lactate? And does that is that difference the risk in developing?
I'm not aware of any data that compared women who are breastfeeding to those who are not breastfeeding. But I'm very well aware that there is several case reports out there that, you know, oxytocin is given to women after birth to stop bleeding, to induce contraction of the uterus. And there is several case reports out there that women with long QT or previously undetected long QT, they go into ventricular tachycardias due to the oxytocin, because oxytocin is giving really, really frequently in obstetrics. So this is why one would recommend a woman with long QT syndrome not to be given oxytocin, because that induces arrhythmias, of course.
Oh, okay, yeah. And I don't know whether it's not a question permitted. Okay, then then at a mechanistic level, you looked into this HERG, and how does it work? There are some data around that estrogens may act on as a direct inhibitor, an allosteric inhibitor of HERG channel, but how does it work for the other hormones? Is that known? Can you speculate on that?
We can speculate, and it's a complicated issue. Estrogen does work on the HERG channel, but it also increases KVLQT1 or KV7.1, which is the other channel. And this is not a homogeneously distributed channel throughout the heart. And one of the main problems with main causes of arrhythmia is always inhomogeneous repolarization. And if you have inhomogeneous repolarization and you increase that by enhancing the channel that is actually inhomogeneously distributed throughout the heart, then you get an increase in arrhythmias. It does also feed back on calcium channels, and there is an increase in LTAP calcium inflow with estrogen, which is not with testosterone or progesterone. So there's a lot of things going on. What actually turns the trigger, that's difficult to analyze.
subject for further study then.
Yes, a lot of further
yeah.
studies.
So I'll give the word back to the pro-actor.
Thank you very much, Professor Biese. The next opponent is not here in the aula, but he is present online, and we see him on the screen already. Professor Stefan Franz. He is Professor of Cardiology and affiliated with the University Hospital in Würzburg in Deutschland. Professor Franz.
Hello, Michael. Good to see you. Oh, I can't hear you right now.
Can you hear me now?
Yeah, now it's perfect. So great talk, of course, and great papers you published there. So I want to ask you some more fundamental questions. So first, you already started on alluding why you used a rabbit and not a mouse. But are there also mouse models of long QT syndrome, and do they help to decipher really what the mechanisms are and what potential treatment could be done? I mean, you already alluded to that those mechanisms are not identical to the human situation, but maybe we can learn also something out of it and not go through the hassle to make these rabbits for years to have at the end one paper.
Highly respected opponent. The mouse indeed was the first model we worked on, and it has advantages, of course, financially. It has advantages of being easier to manipulate genetically, but it has major disadvantages in terms of the currents that they use, the currents that the mouse had that are used for the repolarization of the action potential are completely different from those in the rabbit and in the human. And the mouse also has the major issue that its size is so small that it has no real polymorphic ventricular tachycardias as we see them in humans or in rabbits. You need a special size to have these arrhythmias running. And I know of a lot of mouse models with a prolonged QT interval, but none of these models nearly replicates what we've seen in the humans or in these rabbits. So they're good for very, very basic proof of principle studies, for studies that look at channel trafficking and things like that, but they're not good to go somewhere close to what is seen in humans.
Mm-hmm. Then you say the mutations you made is due to human mutations you found. So are there, how many different mutations are there in humans? Are there a lot? And what made you actually pick exactly this phenotype and work so long with exactly this phenotype then?
The mutations we've chosen at the time we started out were the best characterized mutations that are. There are actually hundreds of mutations out there. And we were lucky to pick, we really had luck to pick ones that create a nice phenotype. And for each of the long QT syndrome, there's really, really hundreds of mutations, and they create sometimes very different phenotypes. They create different problems, causes for the channel. Some cause the channel not to be trafficked to the membrane. Some cause the channel to be dysfunctional in the membrane. And they are very, very difficult to put together in a single model. So basically what we have is we've chosen, and that's also financial constraint, we've chosen these mutations because at that time we thought this is going to give a nice phenotype in the rabbit. And if it would, of course, be tremendously expensive to create more models of all the mutations that have been published so far.
So, but then what are your thoughts on the treatment issue? Do you think that different mutations will have similar reactions to the same treatment because you lose like nicorandil and hormones and so on? Or would you propose that those might be different between different genotypes?
It is very likely that the standard treatment is different for different mutations, and we've done this in a short QT model where we looked at beta blocker effects on the channel properties, and we looked at different mutations for the short QT syndrome and different beta blockers, and it was different defined by the mutation, the effect of the beta blocker. So it's very likely that also beta blockers and long QT are differently effective, and that's actually clinically proven, and that it depends very much on the mutation. The very elegant idea of the last slide I showed of the Ackermann construct is actually that it sort of silences the whole gene, irrespective of where the mutation is, and it replaces it with a functional gene. And if this works, then this would be sort of a gene-specific treatment, which is not mutation-specific. So you could replace the whole channel, but this is far, far, far away from being clinically useful right now.
So when you look at the phenotype of the rabbits, you've seen that in the mortality was especially increased after 1 year. Does this resemble the human phenotype? Because I'm not sure that all of them die when they are 16 or so, yeah?
In humans, we see an increase in the arrhythmias during puberty. We see arrhythmias before puberty, but we see a marked increase during puberty, and we see an increase in long QTs depending on the menstrual cycle of women, and we see it depending on pregnancy. So there is a hormonal influences, but it's not hormonal is not everything in this case. So we see other effects as well. And what was probably the most amazing thing is we have a very inbred strain of rabbits. These are New Zealand white rabbits which have been bred in the same strain for ages. And even though they are very, very inbred and very genetically homogeneous, we see markedly different phenotypes between the siblings of a litter. So you have one litter with very, very different phenotypes. We have nearly normal-looking transgenic rabbits, and we have transgenic rabbits with extremely long QT intervals. And those with extremely long QT intervals actually are those that die off early, and that brought us into problems because sometimes we had trouble keeping the colony. But there are other influences within the heart, within the ion channel trafficking, the ion channel expression, I don't know, where that influence actually the phenotype besides being the very same genotype. And we know that from humans. In humans, we see families which have one mutation, and some of them have symptoms, some of them have ECG signs, some of them look normal from the outside, but still have the mutation. So there's other things going on to balance the ion currents or to interfere with the long QT that we don't know of right now.
So another more clinical question. In the clinical part, we often see long QT syndrome through medications for certain antibiotics, for example, that make long QT, or psychiatric drugs. Is something similar reproducible in rabbits? Or so meaning, do you need the whole knockout, or can you do it by a simple medication to get a rabbit into a long QT syndrome?
This is a very good question.
But you may give a short answer to this question.
And actually, you can induce a long QT in a rabbit as you can do it in humans, and you can use it to study effects of the acquired long QT in the rabbits, which we actually did.
I guess my time is up. That's what it means.
That's right.
Is it like this?
Time runs fast. Thank you very much, Professor Frans. The next opponent is Professor Harry Krijns. He is Professor of Cardiology at Maastricht University. Professor Krijns, het woord is aan u.
Thank you very much, Mr. Prorector. Dear candidate, we've been discussing a lot during the DOSCAM course, and I'm very honored to rediscuss here at this occasion of your thesis, your, I must say, wonderfully prepared thesis, and congratulations on that. Also,
thank you.
The nice presentation today. It was as if I was again at DOSCAM. But also my congrats to the promotion team, Professors Zuhausen and Sluimer. This is a very well-written thesis and pleasant to read for the topic, obviously. It deals with an exciting topic. You're now the 6th PhD candidate from the DOSCAM course, which I'm very proud of. And I must say, I also thank you very much for all the work you did to come to this point. As you know, I'm very fascinated by ECGs, and you showed a very nice ECG of one of your patients, and that's an ECG I'd like to discuss, because there are things in the ECG which are mostly overlooked, because if you see an ECG with a terrible problem, you always look in the middle of the problem. And then we forget to look at the initiation. And you're talking about the short, long, short. And that's okay, that's the initiation. But I was curious what you think of the following. If you now look at the ECG, it's on page 12. And if you then look at the long interval, the first long interval in the tracing. Can you go back? Maybe that would be very nice. So, and take out the running, because you don't need a ECG. So many slides, very nice slides. Then if you take the very first interval, would you agree, it's in the middle now, that that's due to a dropped P wave? So in this case, there is an SA block, because if you measure this, then it's exactly twice the interval. And an SA block then leads to a nearly fatal arrhythmia. Would you agree that this is a dropped P wave?
It looks like this.
Yeah, okay.
The case is a little bit more complicated of this patient, because he not only had a long QT syndrome, but he also came in actually with a severe perfusion problem of his bowel. And actually what brought him to vomiting, and so he was hypokalemic and dehydrated. And this
I don't see the hyperkalemia here.
Yeah, but that's what the sort of main reason was that he came in the first place. He thought.
Yeah, but that's not a hyperkalemic ECG. He may have been hyperkalemic, but this is not an hyperkalemia ECG because it developed slowly, probably. Because if you develop hyperkalemia quickly, then you get these white QRS as well.
Now, what I'm aiming at is that maybe his bowel symptoms caused sort of a vagal reaction, and that therefore.
Okay, that's a good point. But I don't see the patient being vagal, because it's sinus, rhythm, 80 beats per minute. That's not very vagal. Well, but it could be discordant on the SA conduction rather than the sinus node frequency. Might be the case. But okay, forget about this. I don't know either. But it could be a fatal SA block, right? Because you get the first long pause, so you have a short, long, and then all of a sudden you have this. Let's Let's look at the second ventricular premature beat. The second in the number 8 or so. Would you agree that the ST segment of this ventricular premature beat, right in the middle, not the one causing the torsade, but the one before that, that the ST elevation is disproportionate to the QRS width or the area under? Now my question is, why do you think it's disproportionate, and has this something to do with the torsade? Let's try.
That's maybe speculation, because if you think it's disproportionate, you would assume that there is ischemia behind that, likely.
Not this, there's no ischemia. Well, I wouldn't diagnose ischemia on a broad QRS, which is not coming from the conduction system. Because in the previous conducted beats, it's a normal ST segment, isn't it?
Yeah. At least that's what you can say from a ECG that's taken from a monitor, which is not.
Let me ask you something. How long do you think the refractory period in the Purkinje system is? And let's say at the time you see this ventricular premature beat. It could be a premature beat which you introduced with premature simulation. There's still conduction. How long is the refractory period, do you think?
The refractory would likely be shorter than the T wave. Or like the middle of
otherwise, you would not see the beat conducted, right?
Yes.
Yeah. Okay. That's correct. But now I calculated it's 480 milliseconds. relative refractoriness. So it's not the absolute refractoriness. But does this refractoriness hold for the whole heart? That's now my point.
It probably does not. I mean, because that's if the heart was completely refractory, then you wouldn't get the torsade afterwards. So you need to have parts of the heart that are refractory and parts that are not refractory. So for this beat, probably the heart was refractory, but for the next beat, it was not. And this is what triggered the torsade.
I think we are, the both of us, are in the right direction. Because this broad beat, this ventricular parameter beat, with this disproportionate ST elevation, it comes at a time that part of the heart is repolarized. That's why it's depolarized, because you can't depolarize. But the other parts are not repolarized at all. They are inexcitable.
Yeah.
So you get sort of what you can see in ischemia, but it's not ischemia. In ischemia, you have cells which have a very lowish action potential and normal action potential. And if you have them at the same time, then you see the ST segment elevation. So that's the cause of ST segment elevation in transmural ischemia is that you have these ischemic cells which have short, blunted, small action potentials, and you have the normal current is the larger, and this gives this injury current. And in fact, this is not an injury current, but it's a current, and the current is disproportionate because a large part of the heart has a much longer refractory period than these 480 milliseconds. That's why it's coming there. I think, but you might disagree.
I find a hard time disagreeing with you.
Now, you told us the torsade ends spontaneously. What's your favorite explanation for torsade de pointes terminating spontaneously?
Well, for torsade to run, you need changes. As you said, you need refractory parts of the heart and not refractory parts, and these need to be changing continuously. So you need to have areas that are refractory, and when the next beat comes around, they are not refractory anymore, where other areas are refractory. So to keep the torsade running, you need always the interplay of excitable versus non-excitable tissue. Otherwise, you would get a monomorphic tachycardia.
So by chance, it stops by chance.
It stops more or less by chance because it runs into a refractory area.
Why? But why doesn't ventricular fibrillation, so the normal ventricular fibrillation in the setting of acute ischemia, not stop spontaneously? Mostly not. Almost never.
It sometimes does, actually.
Yes, but forget about those cases. So all the other, they don't. Now, my favorite explanation is that torsades, they like long intervals, right?
Yeah.
And they themselves have very short intervals. So the rapid activation of the individual myocytes makes them shorten. And then the overall shortening of the refractoriness in all of these cells makes them go into the direction of the normal ventricular fibrillation, then it may stop. And in the I didn't check that, but near the end of the torsade, the cycle length may shorten. And then, at least that's my favorite explanation. Let's forget about this electrocardiographic exercise.
But Professor Kreins, I have to interrupt you.
I'll accept that. And Floor, give the word back to the prorector. Thank you very much, and thank you for your answers.
Thank you very much, Professor Kreins. The next opponent is Professor Allard van der Wal. He is Professor of Clinical Cardiovascular Pathology at the University of Amsterdam. Professor van der Wal.
Thank you. Dear candidates, it's a pleasure for me to have the opportunity to discuss your thesis. And I can say you have written a beautiful book, layout, it's perfect, and also the contents with this attractive rapid model, transgenic model, which has proven to be useful for investigations regarding also the several publications in highly ranking cardiology journals. So, first of all, I want to congratulate you with this, and of course also your promoters, Professor Suurhausen and Sluimer. And well, personally, as a pathologist, I know very much about the ins and outs of all these this electrophysiology, but my main interest clearly in the field of all these ion channel diseases is great, and that applies mostly to the sudden death, which is also one of the measures of outcome in all your studies. And at autopsies, it's a difficult issue to investigate, because it's said these ion channel diseases, they have structurally normal hearts, which is not really true, at least when you do investigate them carefully in a research setting, as you could also do in your studies. But another thing is that there is often concomitant other diseases which are very prevalent in the human population, coronary artery diseases, hypertension. And my question, how is that from a clinical point of view? Are these diseases interacting with each other? Coronary artery disease, for example, and these ion channel diseases? Even more when I read your statement or proposition number 6, phenotypes of the same mutations, LQTS is variable and depends on unknown factors. Issue also discussed with Professor Frans today. And could it be that relates to an interaction, trigger substrate reaction or alterations in the cellular composition that influences the outcome of these ion channel pathology? It's clear what I mean. And second, does your model provide a tool to investigate these things?
Highly respected opponent. I think what we know clinically is that patients with long QT syndrome, they have their arrhythmias already at an age when usually coronary artery disease, atherosclerosis does not play a role in their life. So up until the age of 30, 35, we know that sudden cardiac death is mostly some form of an inborn genetic disease, either hypertrophic cardiomyopathy, right ventricular dysplasia, or long QT syndromes, or similar entities. And coronary artery disease usually kicks in after the age of 30, 35, depending on your lifestyle, of course. And as far as I know, there is no studies that link atherosclerosis or long QT or the QT-related diseases to this at all. There is I'm not aware of any data that see a connection between these two things. However, we do see patients which have long QT, and we see them sometimes also in older age. And one of the patients I'm following up was an elderly gentleman who was turned 75 when I saw him, and he had never experienced any arrhythmias in his life until he got sick with pneumonia, and he was treated with a substance that prolonged the QT interval, a fluoroquinolone, an antibiotic. And that's when he had torsade points over many hours, always recurring. And that actually triggered the search in his whole family to look for the genetic defect. So it might be that some form of coronary artery disease ischemia might play a trigger in later life, but I'm really not aware of any data that is out there.
And as you say, it's not of interest to go to investigate it. It's not.
It has not been investigated, and I think the rabbits are not a good model because the rabbits normally don't develop atherosclerosis. You have to feed them with high cholesterol diet. And
well, coronary artery disease is of course one, but also hypertension could be. There are some studies done in Amsterdam by Dr. Remme. That was not in the setting of long QT syndrome, but SCN5A mutations in mice, mutant mice, and also in pedigrees of patients with Brugada syndrome. And in these settings, there was a significant increased sudden death in the mice and men of the hypertension group.
Yeah.
But of course, it's another ion channel disease, and also the age is probably can be higher in the Brugada syndrome patients, I believe.
Yeah.
Is that true or not?
Brugada syndrome does have its peak in the early adulthood, usually, as a reason for sudden cardiac death in humans. And Brugada syndrome would actually be a really interesting model to do it in rabbits, because there's no Brugada model out there except for the mouse. And you heard me talk about the mouse. I don't think the mouse is a very good model at all, but it's the best that we have right now. And a Brugada rabbit would actually be very interesting in many aspects. And Brugada syndrome is right now a relatively dynamic field where a lot of things are going on with antibodies and ablations and many different ideas on how to treat Brugada syndrome. But the channel is different, and the dominant negative approach that we use cannot be done with the sodium channel. So it would be something for a CRISPR-Cas model, I think. And so far there has been no CRISPR-Cas rabbit model out there. There might be a reason for that.
Okay. So for the time being, there's no indication to do any studies on the combinations of these diseases.
I think hypertension would be an interesting part, definitely. Hypertension would, and that could also be done. That would be very interesting.
It could be applied in your model also.
Yeah, definitely.
Can I go on? Finished? Yeah. Okay. Thank you very much for your
Thank you very much.
Yeah. Thank you, Professor van der Wal. The fifth and last opponent is Dr. Theo Fei Li. He is University Assistant Professor in Cardiovascular Pathology at Maastricht University.
Yeah, dear candidate, first I will congratulate you for your great work, and I read your thesis with great interest. It's quite new for me because I have never worked with animal model. So my question is especially for the chapter 3, and I find it quite interesting to create a rabbit model. The first question is you created two models, LQT1 and LQT2, and for LQT2 you can show that arrhythmia and sudden cardiac death in LQT2 rabbit, but in LQT1 we can induce or observe sudden death of arrhythmia, but in human that is well a risk factor, it's a very increased risk. What is the explanation about this discrepancy?
The explanation is actually that men are not rabbits, or rabbits are not humans, and that what we see in the rabbits is, as opposed to humans, we see the differences in the regional importance of these channels. So if you have as I said before, if you have a homogeneously prolonged action potential throughout the heart, then you don't have a chance to create arrhythmias, because the heart is either refractory or not refractory. Once you have inhomogeneous repolarization with a long action potential, which we usually have at the base of the heart, and a much shorter action potential at the apex of the heart, then you have differences in repolarization, and this is when the arrhythmias can occur. And the ion channels are there. We have the same ion channels in the rabbits and in the humans, but the distribution throughout the heart is not identical. Okay. So this is the difference between humans and rabbits. In rabbits, the IKS is not as important, and so therefore the differences occur.
Okay. Yeah.
It, of course, would be a larger model would even be better, but the next step would be transgenic pig, and that's another cost issue, I think.
Yeah, it's not processed as a copy of human, of course. But in human, LQT1 can be triggered during exercise and emotion stress. Do you think that if we add exercise stress test in the LQT1 rabbit can trigger the arrhythmia?
We did not do it in vivo, but we exposed the cells to isoprenaline, and we exposed whole hearts to low-dose isoprenaline while on a Langendorf perfusion, and there we could actually see differences in the occurrence of arrhythmias and inducibility, but we didn't see it spontaneously. And this is sort of always an artificial system because you lack the innervation of the heart, which plays a major role in stress and arousal and exercise.
Yeah. And the next question is, in the chapter 3, in that experiment, in 2 of the experiments, you only use the male rabbit for observation of the first one is observation for the awake or free-moving animal. The second one is the observational study for the reason of the sudden cardiac death by the rabbit. In these 2 experiments, you only use the male rabbits and not the female rabbits. What is the reason of that?
We had a resource issue. We had several resource issues. One is that we lost a lot of rabbits due to spontaneous deaths, so we had to keep the colony at a decent size, and the housing of a rabbit per day costs €5 in our facility, and it takes to generate new rabbits, you have to wait 4 to 5 months. So you need to have a large colony of many rabbits because some of them die. And we were, due to financial constraints, we were several times at the point where we nearly lost the whole rabbit line because we couldn't afford to have that many rabbits. So to keep things simple, we sometimes said we'll just test the males or just the females, because if we start making big groups, then you need huge amounts of rabbits.
Okay, next question is also for me is interesting. For your in vivo study, you use isoflurane as anesthetic drug, but the drug itself is also a IKS blocker. You also mentioned that in your article, but the IKS and IKR channel is one of the objects for your study. Why you choose for this drug?
We did one of the papers that is not in there. We actually did an anesthesia study trying to find out which anesthesia does not affect the QT interval in the rabbits. And for the first few studies we did with isoflurane, but we later switched to ketamine, and in combination with xylazine. I'm not aware of the generic name of that, which was the only drug combination. We tried different things. We tried propofol, we tried benzodiazepines, we tried different things, and it was the only combination that was not affecting the QT interval in vivo in these rabbits, and therefore all the further studies are done with that. We actually wrote a paper on that too.
I also don't see any influence on the results, study result, but. Still seems still dangerous use such a medicine for which is the same objective of your study. So
It's not a lot has been known about treatment of anesthesia of rabbits and things like that. This was all things that we had to find out ourselves. As not being veterinarians, and the veterinarians, they don't care about the QT intervals. So we had to find that out on our own.
Another question is for chapter 5. In that study, investigate the potential role of nicorandil in treatment of LQT1 syndrome. But the medicine is ATP sensitive channel opener. It seems to me that there is no direct association with IKS and IKR channel. But you only test the medicine for LQT1 syndrome, not LQT2. But in LQT2, you find a lot of mortality in the rabbit. What's the reason you only choose for LQT1?
The reason why we choose nicorandil, because there's case reports from Japan actually, where they had neonates treated with incessant QT syndrome and torsades. They had them treated with nicorandil and it actually stopped the torsades. So we wanted to replicate that on our model. And the reason why we use long QT1 was, again, our long QT2 line was at some points in time so thin, we had so few animals that we could not afford to use them on experiments.
Okay. Do you think that will work on LQT2?
It probably will work on LQT2 as well. I mean, it is independent of IKR and IKS, okay. So it should be working with no matter what defect there is.
Okay. Yeah.
Mr. Brunner, as you see and hear, the time for defending your thesis has passed. The committee will now withdraw to discuss the quality of your thesis and in particular the quality of your defense. And please await our return here.
Thank you very much.
The PhD defense has now ended. The degree committee will debate the candidate's performance behind closed doors. This process usually takes about 10 minutes.
Southside, 7 miles in my rearview mirror. I know what it felt like. My goal's only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and that. All your time, 'cause I'll go the extra mile. Long road to the south side, 7 miles in my rearview mirror. I know what it felt like. My goal's only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and that. On the dash, got my laces tied. Long road, I don't waste no time. Break rules because fate decides. With the team and we chase the light. I make a move, fall down, shake it off. I hate to lose, bad branch, break it off. No room for negativity, praise and love. Prepare for deep part cause we're taking off. Take the mileage, traveling so far from home. Gathering knowledge, other connections. I put the time in to get everything I ever hoped for. It's a challenge and we all in. It's a better future that we go for. Even though these nights seem long, I know that I am never on my own. Know you've won the battle once you've grown. Southside, 7 miles in my rearview mirror. I know what it felt like. My goal's only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and that. Long road to the south side, 7 miles in my rearview mirror. I know what it felt like. My goal's only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and that. Years can't fly by now, don't waste all your time. 'Cause I'll go the extra mile.
Please be seated.
Mr. Brunner, the degree committee has assessed the quality of your thesis and in particular the quality of your defense. And in view of its positive verdict and of course taking into account the previous qualification that you have obtained, the degree committee has decided to grant you the degree of doctor. And Professor Zuhause is authorized to confer upon you this academic distinction in accordance with Dutch university custom and law. And I invite now your supervisor, Professor Zuhause, to take the floor.
Do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent, independent, and responsible?
I promise.
By the authority vested in us by law and in conformity with the decision of the committee here present, I hereby confer upon you, Michael Brunner, the degree of doctor and grant you all rights attached by custom and law. As evidence of this, I now present you with a degree certificate signed by the rector, the secretary, and the other members of the committee and affixed with the official seal of the university.
Thank you very much.
So, it's my very honor to share some words with you, on you, because this is a product of a long-standing scientific cooperation, not only scientific cooperation, but also a long-standing relation our families have. And I have some years written down here just to the milestones of this work, and I think it started in 2006 or end of 2005. I think first your daughter was born, then my daughter was born. We finally met via the daycare for the children, and we had lots of dinners and hikings and whatsoever. We did social activities, and once I was sitting some years later, 2008, complaining about a person who was unfortunately a level above me and stating about acute loss of memory about a contract we made because I had a grant with a resident for 3 years, which I returned for a PhD student for 3 years. But unfortunately, after 2 years, nobody could remember, and it was my first lesson that everything should be fixed on paper. So at that time, actually, we were sitting there, and you told me that you need somebody to clone your construct, and so it happened to be that of my PhD student, Deepa Pantulu, a highly talented molecular biologist, was able to do the work, and you financed her the last year. So that was a very good agreement, and we're very happy to see the success in 2 papers of this. And so that was the beginning of this scientific cooperation. But it took some years more, I think 10 years, to the 2019-2021 edition of the DASCAM here in Maastricht, which you participated, and it took Harry Kreins to stimulate you to and suggest to perform a PhD thesis here, which you did finally today. And it is a great honor for me to be part of this project. For all of us, I think we're all impressed about the high quality of the research. It has been several times named in the discussion about the quality, about the words like excellence, and so this is a landmark for sure for this topic and for this collaboration. And I wish you and your wonderful family and us a wonderful future. And please keep on the science. And I'm very thankful that I was allowed to be your promoter, and I think I speak also on the terms of behalf of Judith. And I'm also very thankful for your friendship with your family. Thank you so much, and good luck.
Esteemed Dr. Brunner, dear Michael, it's my pleasure to congratulate you with your doctorate, and I do that also on behalf of Maastricht University. And I would also like to share some impressions from the committee with you. Your thesis, we have read it critically, and we have seen that it's an interesting, well-written academic thesis of excellent quality. And we have also seen an impressive list of publications in the scientific literature. And with your respect, I have a number of, with respect to the defense, I have a number of remarks about that. We've got to a diversity and also detailed number of questions. We have heard very clear and appropriate answers. And also you have shown to be very knowledgeable in your field of studies. And we appreciated the interesting, an academic discussion of high quality. And we are very satisfied with your defense. I do not only congratulate you with this result, but also your promoting team, your supervising team of Axel Zuhause and Judith Sluimer. I congratulate you with the result that we have seen today. And also I would like to include in my congratulations your family, your wife and your children and other family members and other, maybe also the colleagues and ex-colleagues that we have that probably have seen, I hope so, to have seen the how is it called, the online viewers. And I would like to thank all the members of both committees, the Thesis Assessment Committee and this Degree Committee, for their critically reading the manuscript and also having an interesting discussion with you. And I would like now to invite Professor Frans to congratulate you, because he's not here, but online we still see him on screen. Professor Frans.
Yeah, Michi, congratulations from my side also. I don't think I have to repeat how excellent you are. You heard that twice already. So I'm very happy that I could be with you. And I think it's a very special procedure here when I compare that to Germany. We have done a lot of PhD defenses. So even at the end, I heard that there will be a group picture with me via Zoom. So I've never had that before. So I think that's a very good location where you got your PhD certificate. So congratulations to you.
Vielen Dank.
Before I close this, thank you very much, Professor Frans. Before I close the academic ceremony, I would like to invite the members of the committee to stand here with you in the middle. And then we will take a picture with Professor Frans still on the screen. And after closing of the ceremony, we will go to the hall, and there we will take another picture at the stairs in the hall. Hereby I close this academic ceremony. So may I invite you to stand with your